Okay, so first I'm going to demystify epigenetics because I'm going to talk about DNA methylation, just to let you know what it is. So we have genes in our genomic DNA, and if DNA is in open conformation, they can be transcribed. If it is in a condensed conformation, they cannot be transcribed. And then if they're transcribed, we have messenger RNA, which is then exported from the nucleus and then translated into proteins, and proteins then function, and that's how we stay alive. So epigenetics is simply that first mechanism that determines whether DNA methylation and histone modifications will make your chromatin open, and you can transcribe it, or it will keep it closed. So why do we need it? Awesome. Why do we need it? It's because the linear length of your genomic DNA, guess how long it is? So in every cell, it is packed into 1 micron nucleus. If I unpack it and just lay it over like that, how long do you guys think it will be? It will be over 1 meter. So we have that much DNA in every cell of the body. So it is then packaged into what we call chromatin. Chromatin allows us to do many things. For example, different cells of the body, such as muscle cell or neuron, get exactly the same book to read, exactly the same genomic DNA, but they read different pages of that book. And that's how our cell fate is determined and is maintained throughout our whole adult life pretty much. In contrast, the way the cells respond to their environment is very dynamic. So epigenetic or DNA methylation changes with diet and also with the weather, if we are happy or unhappy, if it's raining outside or not. So it is not really that we have a stable young epigenome versus stable old epigenome, and we can change one to another, but rather we have healthy dynamics which then become less healthy in a way that is stochastic, even for different cells of the same tissue. Okay, so that was the introduction. So moving on, I already told you in the panel today that there are numerous biomarkers which determine if a person is young and healthy versus old and unhealthy. So there are many, many biomarkers which are different between young and old. So if you would like me to tell you, are you young or old? No problem. We can tell it based on looking at you, measuring your bone density, measuring cholesterol levels, blood pressure, and also looking at your molecules and also looking at your DNA methylation. In contrast, we don't know how do we progress from being young and healthy to being old and unhealthy. So the biomarkers of aging progression remain elusive. And so the general principle of all age clocks is to erase these biomarkers because we don't know what they are and replace them with biologically arbitrary line graph. So in this approach, the young versus old parts of the graph will be roughly the same in real life and in the model because we make line from A to B. Our assumption is that somehow process goes from A to B. We don't care how it goes from A to B, but we just want to connect A and B with a straight line. So then we are in actuality are erasing the natural biological patterns. Let's say that we start with this pattern of DNA methylation where beta values is a probability that DNA is methylated, and this is the age of a person. Then we apply biological clock, also known as elastic net regression. So we do various things, we select various data points, we do weights assignment, and gradually after many iterations, we make a line graph. Now, while we can make this line graph go in this direction, we cannot predict anything biologically important because we replace DNA methylation data with biologically arbitrary numbers. If we did not replace them, it would look like that. But to make a line graph, we have to change them. Some people also are confused or think that we are discovering a pattern because we have some data for model training and some data for model testing. But that is not true because we iteratively do 10 or more rounds of training and testing using different parts of the DNA methylation data sets of different people, such as that eventually we use all the data for training. So what we did, we fitted DNA methylation results into a line graph by performing biologically arbitrary transformations, colloquially known as data fudging. And then, as I mentioned, our results will be roughly repeatable from one human population to another because we make a graph between beginning and the end. But how roughly? So here is the natural human dynamics of epigenetics and different clocks, Horvath, Hannam, PhenoH, AdaptH, DAMH, COSH, shows that the errors could be 60 years. And all clocks, most of them predict that the entire population of people is younger. But the worst part is that they overlap predictions for healthy people in blue and patients with various diseases of inflammation who are in red. So again, I told you I like metaphors. So this is a metaphor of hurricanes in Florida. Imagine that instead of knowing where hurricane goes, we just modeled it as a straight line. Said it starts here, it ends there. I don't care of the path. I'm just going to connect this point. And if you live here, you need to evacuate. But if you live in this area, you're fine. It would not be very productive. So particularly for all of us that are in the path of the hurricane, right? We are in the path of aging. We don't care that we start young and old. We care how we progress. So another thing that happens with biological clocks is that they change the direction of change or biological information with age. So for example, there are things we change with age upward, for example, your weight or blood pressure, and other things simultaneously change downward, for example, bone density. So it's not everything is up or everything is down, right? There is different collinearity. And the same is true about DNA methylation or genome. But because clocks apply weights without regard to the actual change, it is just a machine learning process, then the directionality of information changes, making it even more difficult to figure out what was being predicted. And these errors of incoherence are very high in different clocks and different models. The errors are in orange. And even one error would question interpretability of the clock predictions. So then we can correct these errors and make clocks more coherent, as we published this year. And you see right away, you start seeing who has diseases and who is healthy. But if you look at the histogram width, now we cannot predict your age very nicely in a linear fashion. Instead of, 40 years errors, we start making 70 years errors. So we can tell who is healthy, who is not healthy, but we cannot simultaneously tell you how old you are in a linear fashion because you do not age linearly. So then it was strange to us that clocks that operate on the blood cell DNA methylation fail to detect inflammation, fail to tell who has disease of inflammation, who doesn't. And then the suggestion, and it was actually surprising to me to learn, that suggestions that clock laboratories have is that why don't we remove any heterogeneity in the data set so we don't know what cell it is, and then we build our model. But heterogeneity is the hallmark of aging, of normal aging and inflammation. So cell heterogeneity, if you go from normal tissue to fibrosis, or if you go to normal blood subsets to inflammatory macrophages. Heterogeneity of cells is a reflection of aging, and I don't understand why we should remove it if we are building age clock. Additionally, it's not easy to figure out how to remove it. So we don't know. There is not a gene which tells that cell is a macrophage. It's a combination of many genes. So try to modify DNA methylation databases before building clock models maybe is not very productive either. And so then if you look at this more coherent model, it is not really biased to any blood cell subset. But if you look at the skewed models, which are incoherent, they actually are counting leukocytes. So they're counting leukocytes, but they fail to distinguish who has disease of inflammation, which is a super interesting point, suggesting that inflammation is more controlled by the difference in gene expression than by switching from T cell to macrophage. Okay, so now moving on, what is the solution? If we keep drawing line graphs, we are depriving ourselves of the figuring out what is the natural curve of aging, what it is. To understand that, first of all, we need to have hypotheses, and secondly, we need to keep the experimental data intact. We cannot start weighting it in a biologically arbitrary way. So hypothesis is, as you probably guessed from my morning summer panel, that biological age is biomarked not by the mean level of molecules, but by their noise, standard deviation, or coefficient of variance. So for example, we don't know what exactly is wrong with your car, but there is some noise, and we know that it's not good. And so it's very difficult for us, and in my opinion, impossible to find stepwise biomarker of aging because noise increases with aging, and it will mask any sort of gradual change. So the natural pattern of biological aging was found and discovered from the Convoy Laboratory in 2023, and that's what Generation Lab is founded on. We call it noise barometer, because I don't like clocks, so what other instrument we can then call it that measures something, as pressure of aging and disease on an organism. And here is an example of one gene in your DNA, where each dot is a person, and if you look at the mean levels, they are roughly the same between ages 27 and ages over 70. But if you look at the standard deviation, you see how noise increases with aging. And then you can identify, let's say, 50 of such areas, which are on average the same between young and old people, and ask, is there noise increases and how much? And you get this natural physiologic curve of aging progression, which is not a model. It is direct quantification of unadulterated experimental results. And then here you see that if people have arthritis, the noise increases earlier, and you can say that they're older. Again, this is just quantification. And so we have this pattern of rapid noise increase, short-lived plateau, additional increases. So then how many at all other such areas in our genome? We looked at 500,000, 500,000, and we identified 460 parts of the genome. And our curve, not our, but physiological curve of aging, is remarkably reproducible when we sum up standard deviations. And it can be even seen with a single resolution. This is not even adding them. This is one standard deviation measurement of this one area of the genome through human chronology. So this, guys, how you grow old. So then in Generation Lab, with the great work of Ranting Han, who is a data scientist, we checked if this curve is reproducible for different organ systems and functions, such as cardiovascular, or immune, or inflammatory, or fibrosis, not just organs, but also functions. And turns out that it is, which is remarkable. So here we have 460. These genes encode proteins with different functions, and if you group them by function, so it will be less than 460, then each function will form the same curve of aging or noise increase. So noise increases in a conserved way throughout our body, and that allows us to then have very early detection of dysregulation before there are symptoms. So it was always generally known that we do not age linearly, right? We do not grow 1 gray hair per day, and that's how eventually we have gray hair. But systems age allows us to understand causal mechanisms. What happens at this point to epigenome? What happens here? Why do we leave plateau and so forth? So it is the only and the earliest detector of loss of health and increase in disease risk before there are symptoms and before clinical tests could be tabulated and tell you that phenotypically you're already old. We tell you why. So then with Alina Su and Michael Saswell, who are very successful serial entrepreneurs, we started Generation Lab, and Alina is here. And then what it allows us to do, it allows us to know the efficacy of medicine if it is applied to very early pre-symptomatic health decline. Do we actually have all of the drugs that we need for preventive medicine? We just don't know when to use them, and now we do. And also, we can rule longevity approaches in and out. We can inform people and their doctors. We have many affiliated clinics on the best action plan. And we have novel Generation Lab proprietary database on longitudinal biological age. So noise barometer or systems age is like the early warning system, like you drive a car and you have something flashing in yellow, so you know that something is being broken. And then the last 1 or 2 slides, I will just tell you about what to do about it. Do you have a broad age or health defender? So you probably already know, if you followed some of our work, that blood hydrochronicity could be rejuvenative, and so can be plasma dilution without young blood. So for example, there is reduction in circulating cells with DNA damage or senescent circulating cells, and there is restoration of productive lymphoid to myeloid ratio through plasma dilution in people. And there is reduction of proteome noise of the systemic proteome. So then our quest was to see, can we replace this by a drug? Can plasma dilution be a shot? So then in the Canvo lab, and that is the latest paper that came out of the Canvo lab, we tested it. And so we used 2 molecules. One is called oxytocin, and another is called ALK5 inhibitor of TGF-beta signaling. So TGF-beta becomes overpronounced with aging, causing fibrosis and inflammation and poor regeneration. Oxytocin goes down with age, which also causes poor regeneration and maintenance of muscle, bone, and metabolic states. So what happens if you treat old mice, which already are old and frail? We started them when they're 25 months old, periodically with ALK5 inhibitor and oxytocin. So what happens, actually excellent things for men and not so good things for women. Because in male mice, they lived much, much longer. This is actually much longer than with any other reported approach. So they lived 157.22% longer than control mice. Not 15, not 30, 157. And this is median, right, if I count here in the larger part. But female mice did not improve. And then male mice also had better health span and resilience to death, but female mice did not. And the reason is that when we tested what happens to systemic proteome, in male mice, they responded to TGF-beta inhibition and oxytocin by diminishing all of these pro-inflammatory age-related proteins and calibrating to health these signal transduction networks. And look what happens to females. They were not responsive. And that is despite the fact that initially in randomization, females were not more frail than male mice. They were equally as frail. This is my last slide. So it actually is super interesting to me that not only females and males age differently, but they respond dramatically different to some of the longevity drugs. It points to amazingly interesting mechanisms. And at this point, I switched full term to Generation Lab, and now we are working on our first compounds to overcome this hurdle and to build on the positives. So I would like to shout out big help, thanks to the Canbo Laboratory over the years and to our wonderful funding.